Hello students, in the last class we were discussing about cell structure and its functions, and we have also discussed about the nucleus. What is nucleus? What is its functions? And who named the nucleus? We have all discussed in the previous class. So let's revise once again. So when you observe a nucleus in a cell, it is the most clear and the distinct part that you can see. Further, the name nucleus was given by Robert Brown, whereas another scientist, Schleiden, has named the nucleus as cytoplast. A nucleus does the functions of storing the genetic material. Okay, and it also has the functions of controlling and regulating all the activities of the cell. For example, nucleus helps in cell division. Nucleus also helps in regulating all the activities of the cell. Okay, all the genetic information that regulates a cell is stored in the nucleus. That is the reason why even hereditary factors, say for example, whatever hereditary factors like hair color, eyes, or your characteristics, whatever physical characteristics or any other traits that you see in yourself is transferred from your parents because of the information that is stored in the nucleus. Okay, so the nucleus stores the genetic information of the parents and transfers it to the children. This is how the nucleus is seen under a microscope. All the characteristics of the parents are transferred to the children because of the genetic material that is present in the nucleus. So, the function of the nucleus is to store the hereditary information, to regulate the functions of the cell, and also to help the cell in its division. So, there are two kinds of cells depending on how organized the nucleus is. Now, when you observe a prokaryotic cell and a eukaryotic cell, in both the cells there is nucleus. But when you observe the nucleus in a prokaryotic cell, it is irregular. Okay, it does not have a definite shape. It does not have an organized nucleus. But if you observe the nucleus in a eukaryotic cell, you will find that it has a definite shape. Okay. So, a prokaryotic cell is a cell in which there is nucleus, but it is not properly organized. So, the absence of an organized nucleus is characteristic in a prokaryotic cell. For example, in cyanobacterium, you find the nucleus is not properly organized. When you look at a eukaryotic cell, you will notice that the nucleus has a definite shape. Okay. Example, in all animals and plant cells, the nucleus is very well organized. Hence, it is called as a eukaryotic cell. And do all cells have nucleus? No. Red blood cells and the sieve tube cells of the plants do not have a nucleus. In all mammals, in red-blooded creatures, the red blood cells in human beings and in other animals do not have a nucleus. In plant cells, especially in the sieve tube cells, the nucleus is absent. So, not all cells have a nucleus. Let us move on towards cytoplasm. Even cytoplasm we have discussed in the last class. So, cytoplasm, when you stain onion peel cells and mount them on a microscope, you will notice that there is a darkly stained areas which is the cell membrane, and there is a lightly stained areas. These are all very light blue stained areas. This area or this part of the cell is called as the cytoplasm. Cytoplasm is always enclosed in a cell membrane or a cell wall, okay, which is dark blue in color. So the light blue part of the cell is called as the cytoplasm, and it is always fluid filled, okay. It is always filled with the fluid. Now this fluid contains or holds all the cell organelles within itself. So this is the blue color area is called as the cytoplasm, and in the cytoplasm we observe that it has many organelles like nucleus, you have endoplasmic reticulum, mitochondria, you have Golgi bodies, and you have different lysosomes, and all are floating in the cytoplasm. And the cytoplasm is always enclosed in a cell membrane or the plasma membrane, okay. And then the fluid which is inside the nucleus is called as the nucleoplasm, and the fluid which is outside the nucleus is called as the cytoplasm. Now let us move on towards ribosomes, endoplasmic reticulum, Golgi bodies, and lysosomes. Today we are going to cover 4 topics. One is ribosomes, endoplasmic reticulum, Golgi apparatus, and lysosomes. I want you all to observe the structure of the ribosomes carefully. Now in this picture you see that there is a nucleus, there are some tubes around the nucleus. On the tubes you find that there are small granule-like structures which are red in color. You see these small granule-like structures which are scattered on the tubes. These are called as the ribosomes, okay. These small granule-like structures are called as ribosomes. Now why are these ribosomes present over here on the endoplasmic reticulum is, ribosomes, wherever ribosomes, the red granular structures that you see here, wherever these structures are present, over there proteins are formed, okay. Wherever you see the red granular structures called the ribosomes, ribosomes help in manufacturing proteins. So that is where protein synthesis happens. And you will notice that these granules are made up of RNA, okay. How are they made? These ribosomes, how are they made? They are made of RNA. When 2 RNA are combining together, they form a ribosome, okay. So when you observe a microscope, under a microscope, when you observe a cell under a microscope, you will find that these ribosomes are dark black in color. You see these black granule-like structures? These are called as the ribosomes. And wherever ribosomes are present, in that area proteins are synthesized. Proteins are manufactured, synthesized, produced, okay, all are the same words. So these black structures that you see, granule-like structures, are called as ribosomes. So ribosomes are granule-like structures, they are found in cytoplasm. They are formed of RNA proteins. I was saying, these ribosomes, how are ribosomes formed? They are formed of RNA molecules. When RNA to RNA combine together, they form ribosomes. There are 2 types of ribosomes, free ribosomes and attached ribosomes. They are the sites of protein synthesis, as I said. Ribosomes are of 2 types. Some ribosomes are freely floating in the cytoplasm, whereas some ribosomes are sitting on the blue tubes. So the ribosomes which are freely there in the cytoplasm are called as free ribosomes, and the ribosomes that are attached to the endoplasmic reticulum are called as attached ribosomes. And wherever ribosomes are present, wherever these granule structures are present, over there proteins are synthesized, proteins are manufactured. Now let us study endoplasmic reticulum. I want you to look at the structure of endoplasmic reticulum. Now when we were studying ribosomes, we have seen that these blue color tubes are present around the nucleus. Around the nucleus you have found that there are some blue tubular structures that are made up of membranes. These blue tubes are called as endoplasmic reticulum. And they are present around the nucleus. Observe, look at the structure of the endoplasmic reticulum. You will find that they are present around the nucleus. And in this structure of endoplasmic reticulum, you find that there are two types of tubes. In one type of tube, you have small granule-like structures, which we said ribosomes. And there are tubes in which there are no ribosomes. So, these structures on which ribosomes are present is called as rough endoplasmic reticulum. On these tubes, when there is no ribosomes, these are called as smooth endoplasmic reticulum. Okay. So, now wherever ribosomes are present on the tubes, these are called as rough endoplasmic reticulum, and where ribosomes are not present on the tubes, it is called as the smooth endoplasmic reticulum. Now, what is the function of endoplasmic reticulum? It transports the substances like sugars, proteins, molecules, all are transported from the nucleus to various parts of the cell through endoplasmic reticulum. So, it transports substances that are manufactured in the cell from one part of the cell to the other part of the cell. So, it acts as a transport system. This is how endoplasmic reticulum is seen from a microscope. When you observe a cell under a microscope, this is how endoplasmic reticulum is seen. Now, the structure on the left side, this is the rough endoplasmic reticulum because it has ribosomes sitting on them. You see, many ribosomes are sitting on the tubes. So, this is called as the rough endoplasmic reticulum. When you observe the structure here, you see that there are no microsomes, there are no ribosomes, and the tubes are very smooth. So, this is called as the smooth endoplasmic reticulum because it does not have granules like ribosomes on them, right. So, this is the rough endoplasmic reticulum, this is the smooth endoplasmic reticulum. So, as I said, endoplasmic reticulum is a network of membrane tubes. See, you see how many tubes are there? So, these tubes are called as endoplasmic reticulum, the tube-like structures. Okay. What do? They transport substances from one part of the cell to another. What do? They transfer all they transport substances from nucleus to other parts of the cell. So, it acts as a network for transporting substances. And endoplasmic reticulum have granule-like structures on their surfaces. These are called as ribosomes. As I was saying, these granule-like small tiny structures that you see on the surface of endoplasmic reticulum, these are called as ribosomes, and these tubes on which ribosomes are called as rough endoplasmic reticulum, and if the endoplasmic reticulum does not have any ribosomes, it is called as smooth endoplasmic reticulum. So if the tubes have ribosomes on them, it is called as rough endoplasmic reticulum. See, if these endoplasmic reticulum have ribosomes on it, it is rough endoplasmic reticulum. If it is smooth without any ribosomes, then it is called as smooth endoplasmic reticulum. So, rough endoplasmic reticulum helps in manufacturing proteins. Smooth endoplasmic reticulum helps in manufacturing fats or lipids. And both proteins and fats help in building the cell membrane. Okay, so when you see these structures, rough endoplasmic reticulum helps in making proteins. Smooth endoplasmic reticulum in a cell helps in making fats or the lipids. Okay, so the proteins that are made out of rough endoplasmic reticulum and the fats that are made of smooth endoplasmic reticulum, proteins and fats, these two things help in cell membrane, preparing the cell membrane. And what is the function of endoplasmic reticulum? It transports substances from the nucleus to other parts of the cell. It also helps in making proteins and lipids or fats, and it also helps in detoxifying the poison substances from the body. So wherever you see this endoplasmic reticulum, especially in liver cells, it will help in removing the poison out of the body. Okay, so the smooth endoplasmic reticulum on the liver cells helps in removing poisons and drugs from the body. This is the functions of endoplasmic reticulum. Now let us move towards Golgi apparatus, Golgi bodies or the Golgi apparatus. I want you to look at the structure carefully. So this is how the Golgi bodies or the Golgi apparatus looks under a microscope. So what are Golgi bodies? They were first observed by a scientist called Camillo Golgi in 1898. He observed for the first time under a microscope, and hence he named these structures as Golgi bodies. So these are sac-like structures that are made of several membranes, and they are filled with many and they are surrounded by vesicles around them. And these are found in cells that secrete hormones. Okay, so what is the function of Golgi bodies? They pack various proteins and substances that are produced by the ribosomes, which are transported to cell membrane or other organelles. Okay, so when you observe the Golgi bodies or the Golgi apparatus, what do you notice? These are sac-like structures. You see that they are like bags. This is a bag, another bag, a series of bags. Okay, when you look at the structure, it looks like sac. A sac is nothing but a bag. So when you look at the structure of Golgi apparatus, they are looking like sac-like structures. And around the sac-like structures, you have fluid-filled vesicles. These are the fluid-filled vesicles that are surrounding the Golgi apparatus. Now, who gave the name Golgi bodies? Who gave the name? A scientist by the name Camillo Golgi. He observed the structure of Golgi bodies under a microscope and gave the name Golgi bodies. Okay, he was the first person to observe the structure and then give the name, and then he gave the name Golgi bodies. Okay, so what do they do? Golgi bodies, now we said ribosomes make proteins, right? We said that these structures, ribosomes, they make proteins. Wherever ribosomes are, its function is to make proteins. So the Golgi bodies packs the proteins that are produced by the ribosomes, and they pack these and transport it to other cell or to other cell organelles like lysosomes or to other cell structures. Okay. And these are found, Golgi bodies are found in cells that secrete hormones. For example, you can find them on pancreatic cells, you can find them on thyroid glands, you can find them on salivary glands. Okay. So they are found in cells that secrete hormones. Now let us look at lysosomes. I want you to look at the structure of lysosomes. Lysosomes are called as the suicidal bags of the cell. What is the reason? This is how the lysosomes look under a microscope. So, lysosomes are tiny particles found in cytoplasm. They contain destructive enzymes. Whatever needs to be digested or whatever needs to be destroyed are transported to the lysosomes. Lysosomes digest these materials and sometimes they burst, releasing the enzymes into the cell, thus digesting the cell. Okay. So, the lysosomes, when you observe the lysosomes, these are very tiny structures that are found in the cytoplasm. Okay. What does a lysosome do? Lysosomes contain a destructive enzymes or enzymes that are capable of destroying anything. Okay. So within a cell, if something has to be destroyed, all those materials will be sent to the lysosome. Okay, if the cell wants to destroy some proteins or if the cell wants to destroy some virus or bacteria, it will send that to the lysosome. Now the lysosome contains the killing properties. It has the property to kill anything. Okay, it contains those enzymes or the chemicals which are capable of killing and digesting and destroying the substance. So when a bacteria or a virus or a protein or any waste substance is sent to the lysosome, it eats it up, it digests it and destroys it. Okay, so the lysosome is capable of destroying any substance. So lysosomes in a way help in cleaning the cell. So whatever waste materials are found in the cell which are of no use to the cell, they are sent to the lysosome department. So the lysosome destroys them or digests them. But sometimes the enzymes that are there in the lysosomes burst and all the poison or all the destructive enzymes come out and they eat up the whole cell. Thereby the cell will also die. The whole cell also dies. So that is why lysosomes are called as the suicidal bag of the cell, because it is capable of destroying, it is capable of completely digesting or destroying the whole cell. So lysosomes are very tiny structures. Tiny means very small structures that you see in the cytoplasm. They contain digestive enzymes within them. Inside the lysosome there is digestive enzymes. Whatever waste materials are there in the cell, they will be sent to the lysosome. So the lysosome takes those substances, it digests them and destroys them. Sometimes it even bursts and then those digestive enzymes come out of the lysosomes and kill the whole cell. or destroy the whole cell. Hence they are called as the suicidal bags of the cell. We will discuss about mitochondria and other cell organelles in the next class. So today we have discussed about ribosomes. In the different cell organelles, today we discussed ribosomes, endoplasmic reticulum, Golgi apparatus and lysosomes. 4 cell organelles we have discussed. I want you to read the textbook, read about ribosomes, endoplasmic reticulum, Golgi apparatus and lysosomes. Okay. Okay. Thank you. Have a nice day and read the textbook. I want you to read the textbook. And if you have any doubts, let me know. Okay. Thank you. Bye.